Hello, welcome to my presentation, Investigation of Plasminogen Activation System Family as Prognostic Biomarkers in Pancreatic Ductal Adenocarcinoma. As we all know, pancreatic ductal adenocarcinoma has a dismal prognosis and novel therapies are urgently needed. In this regard, integrated analysis of accumulated multiomics data could be useful to investigate potential diagnostic and prognostic biomarkers of pancreatic ductal adenocarcinoma, as well as to explore associated molecular mechanism, which in turn could lead to the development of novel therapeutic approaches. In most cancers, plasminogen activation system is known to influence tumor progression. However, a comprehensive expression analysis of components of plasminogen activation system in pancreatic ductal adenocarcinoma progression has not been previously performed. Therefore, we aimed to explore clinical significance and prognostic value of plasminogen activation system family genes in pancreatic ductal adenocarcinoma using bioinformatic approaches. This study was implemented according to the flowchart shown in this figure to elucidate and establish the gene signature and to carry out validation of the prognostic model. We used 6 geodatasets of microarray and 1 dataset of RNA sequence from NCBI comprising more than 470 samples. Plasminogen activation system genes that were differentially expressed in pancreatic tumor versus normal pancreas were identified at absolute log-fold change greater than 1 and adjusted p-value less than 0.05. Then we performed integrative bioinformatics analysis to identify significantly enriched cancer-associated pathways, biological processes, molecular functions, and cellular components related to the identified plasminogen activation system's differentially expressed genes. We also performed network analysis of gene-gene interactions and protein-protein interactions of the plasminogen activation system's differentially expressed genes. In order to validate the expression of plasminogen activation system genes at mRNA and protein level in pancreatic ductal adenocarcinoma, we analyzed cancer cell line encyclopedia, the cancer genome atlas, and human proteome data. Then we performed survival analysis based on both clinical information and RNA sequence gene expression data for plasminogen activation system genes in pancreatic ductal adenocarcinoma from the Cancer Genome Atlas via CBio portal. We further applied a standard Cox proportional hazards regression model to perform univariate and multivariate analysis, considering each gene individually and all genes of plasminogen activation system at a time, respectively. The above methodology allowed us to identify plasminogen activation systems components that could be useful as prognostic markers. Let's see the key findings and conclusion of this study. We have found that plasminogen activation system family genes are significantly differentially expressed in pancreatic ductal adenocarcinoma versus normal pancreas. And these differentially expressed genes are associated with core cancer-related signaling pathways, such as TGF-beta, P53, DNA damage, response, etc. The plasminogen activation system differentially expressed genes are also associated with major hallmarks of cancer, for example, invasion and metastasis, evading growth suppressors, immune evasion, neoangiogenesis, proliferation, tumor-promoting inflammation, genome instability, and mutation and cell death resistance. Our univariate and multivariate analysis revealed that PLU, PLG, SERPIN-E1, and SERPIN-B2 were independent prognostic markers predicting poor survival significantly in pancreatic ductal adenocarcinoma. Further, Kaplan-Meier analysis demonstrated that concomitant high expression of PLU and SERPIN-E1 was associated with significantly lower median survival compared to low expression of both genes in pancreatic ductal adenocarcinoma patients. Thus, our findings imply that plasminogen activation system genes have the potential to be reliable predictors of patient outcome and may aid personalized therapeutic decision-making in pancreatic ductal adenocarcinoma. If you would like to know more, please have a look at my poster or send me a message. Thanks for watching.